Hello and welcome to See You at Kanata. My name is Travis Fleeler and I am the Director of Operations for Carleton University here in Kanata. Our conversations today are going to be meeting with student innovators. So these are the builders, the great minds, the thinkers, the leaders in our classrooms who are gaining skills and applying those skills, whether it's in co-ops, internships, capstone projects, or even working directly with companies on research and even starting their own companies. We want to showcase the type of talent that Carleton has and hopefully can translate that into how they're going to impact industry, the future, and our communities as a whole. So thank you for joining us, and we hope to see you at Kanata. Today, I'm happy to be joined by George Hilan. George Hilan is a biochemist, a podcaster, an all-around science communicator who's helping bridge the gap between lab work, real-world impact. And so George, I want to thank you for joining us today.
Travis, thank you so much. It's an honor to be here.
Yeah, so George and I first met at an event downtown with Life Sciences Ontario. And we immediately started talking about how to work together on really the science communication side of things, which I thought was extremely interesting. But before we really dive into that, tell us a little bit about your journey with Carleton University and getting into biochemistry.
Oh man, it's interesting because I started Carleton long ago. It was 2014, I think. And the first couple of years, I was really just at school because that's the trajectory you take. It wasn't because I was actually really wanting to get into science, but I got into biology at first. And after a couple of years, after 2 years I think, I met Dr. Marta Mullally. Yep. She was a biotech instructor or biotech professor at Carleton. And it was really the way that she taught the courses which made me very interested in science at that point. That was my first time really truly engaging with the content. Yeah. And I also had a second biotech course which two researchers from the NRC came in, Dr. Sue Twine and Kelly Fulton. Yeah. As a result of that, I became interested in research more. We had an internship week at the NRC, and it was a blast for me. That was really my first time being kind of introduced to the world of research, and I fell in love at that point. At which point, I decided to get back to school and just focus on studying properly this time, and the rest is history.
And then you decided to continue that education journey and go and do your master's. Can you tell us a little bit about how you decided and why you decided to continue that journey?
So after the internship week, I got the ability to work at the NRC for about 4 months, the contract, and it got extended again for another 4 months as a co-op student. It was an incredible experience. The research was phenomenal. The people around me, the researchers, were they're very knowledgeable. They know, the ins and outs of the research field, and just learning from them was just an absolute pleasure. So that got me interested to go further down in my education, and so I decided to get into a master's degree. I did it with Dr. Wilmore and David Sabatino, and it was amazing. We had a great project going on, and it made me want to kind of continue down that path.
Yeah, so some students are a little hesitant to do their internships or co-op placements at Carleton because they want to get a varied experience. They're doing their studies here, and they think, oh, I want to go work in industry. Can you tell us some of the pros to sticking around the university and what kind of skills you were able to acquire, even though you were studying here, sticking around and doing your internship on campus? How was that beneficial for your career outlook?
So on campus, the two researchers, Dr. Twine and Kelly Fulton, came over, and I don't know that I would be here talking to you right now if it wasn't for them. And Carleton provided that opportunity in the biotech class, and I felt very privileged to have been able to engage in that research and to be a part of it. And I believe it's really what led me down that kind of journey of scientific research that I became so involved in right now.
So let's go back to the science communication side of things. Obviously, you have an entrepreneurial side to you as well, where you started your podcast, Where Knowledge Stems From, which I found was fascinating to really kind of break down the barrier between science, terminology and communication, and just kind of, defining it a little, better for people like myself who aren't scientists, who don't work in that field, who don't necessarily know, the scientific words for, what you are involved in every day. Tell us about that side of your passion and how that came about.
So it's interesting because during the master's when I did the podcast, it's kind of taken a pause right now because things are very busy, but exactly. But during my master's, I was kind of interested in figuring out whether I wanted to do a PhD or not. And so I started talking to my peers around me, and because everybody's either doing their master's or doing their PhD, I figured they all have some sort of understanding as to why they wanted to get into those fields or pursue those degrees. So I started asking them questions about their interests and their research, and I thought everybody had very interesting backgrounds. Everyone had different paths to how they got to where they were. And I figured maybe there are other people out there that would be interested in kind of knowing this information. Yeah. And so that's kind of where the birth idea of the podcast came from. And I figured I'd invite those individuals on. And it went from graduate students to just researchers because I became more and more interested in the fields of research around.
And working at Carleton, you meet some people.
Absolutely. Oh, you meet a lot of people. Like
it's the conversations you have on campus, they're crazy. The amount of different areas of research that we work on is so impressive. When I was in school, not to talk about myself, but when I was in school, you don't realize the amount of research and work and the amount of people that are involved in kind of making an impact for the greater good, right? And when you are more immersed into the university, you see that, and it's crazy.
I mean, I completely agree with you, because being around those different labs and having access to those professors and researchers and innovators, you realize how much knowledge and how much experience they all have.
Like the amount of industry reps and partnerships that exist within the university, I didn't realize either, right? The university can act as that research and development branch for a business, and that kind of dovetails nicely into NuvoBio, which started at Carleton University. Obviously, where you're employed now, we were able to take the skills you learned at the university and work with an organization that was spun out of Carleton. Can you tell us a little bit about the work that you're doing there right now, and maybe give us a little background on NuvoBio?
Absolutely, yeah. So currently I'm a lead research tech there, and my role is to essentially validate the data that we get from AI. So our computer engineer has developed an algorithm. It's essentially an AI platform that can accelerate the identification of peptides towards any type of protein that you're interested in drugging. So essentially, let's say a company comes to us like, hey, we're interested in kind of developing a peptide therapeutic for this particular disease, and we're interested in this protein. Right. So this AI will mine the proteome for different protein-to-protein interactions. It's a large network, over 1.8 million validated data points. So what it'll do at that point is identify sequences, many sequences of peptides that can interact with this protein. Now, the part that really sets us off from the competition is that while we can do that with high accuracy, however, we can also identify off-target effects. Right. So, you want to drug, let's say, protein 1, but ideally you don't want this peptide to interact with any other proteins, especially if it's not by design. And that's what our platform excels at doing. You might say, why peptides? Well, I'll say because not only are they naturally occurring in the body, and we can make them very accessibly, but they're also immunogenically safe. So our bodies are less likely to to kind of have this immune response to them as you would with other types of medication. Also, they're very cost-effective in manufacture. So when we develop therapeutics that would eventually lead to the market, they're not going to cost an arm and a leg for people to buy, which is huge in the world of kind of disease management and treatment.
Well, pharmaceuticals in itself is just it varies from the U.S. to Canada to Europe, and anything to make it more affordable for everybody else is
exactly
kind of the goal, right? Going back to the storytelling, I was able to see you translate what you were doing with where knowledge stems from podcast, and being able to do a little bit of promotion and marketing with NuvoBio, and kind of explaining what peptides are, what amino acids are, and things like that. As you're kind of working through all of these different videos and podcasts, talk to us about the challenges of doing these podcasts, and what would you say has been most surprising about doing that kind of work?
So for the company, and because I had some background in aspect of video making, I thought it would be very interesting to create some videos that kind of engage the audience, really kind of explain some of the science, make it so that they're aware of what we're doing. Yeah. And that's kind of where some of those videos came from. So what is a peptide? Actually, it was our COO's idea, Valentina, to do the kind of the walk-in, mic talk, ask people questions, where what a peptide is. And that was interesting because we just walked around campus asking people, and some knew, some didn't, and so we had different levels of knowledge of who knew what peptides were. There were other videos where we were communicating what we're doing essentially at the level of whether, it's like the complete workflow, bottom down, kind of from the top to bottom. And it was really meant to just try to explore that video content creation idea and couple it with the science to just kind of put ourselves out there more.
I'm going to pick some everyday objects, a couple of them, and I want you to explain the biochemistry behind it if you can.
All right, let's do it. Let's try it. Let's try.
Let's start with the first one, which would be coffee. How does
This is water, by the way.
Oh, okay. Well, this one is definitely coffee. Can you explain the biochemistry behind coffee?
So the active ingredient in coffee is caffeine, as many people are aware of. And the way it works is it doesn't actually give you energy. So it just blocks the receptors in your brain that make you think that you have energy. Essentially, adenosine. It's from what I understand, it's an adenosine antagonist, an adenosine receptor antagonist, meaning that it binds to the same receptors that this molecule adenosine binds to, and adenosine it builds up through the day after you wake up, and then towards the wee hours of the night, it becomes higher in concentration, and it makes you feel sleepy.
Okay.
So what coffee does is it binds to that receptor instead, and it prevents adenosine from binding and makes you feel like you have more energy or that you're not as tired. And so that's kind of the very high level kind of explanation of how coffee works.
Can a person drink so much coffee that their body becomes immune to it and caffeine really doesn't impact their system anymore?
Oh yeah, I mean, I don't know if you can drink enough for your body to become immune, but you definitely build a tolerance.
Yeah.
So the more you drink coffee throughout the days and the months and the years, your body starts to become more and more tolerant to it, and essentially, as I understand, starts to develop more of those receptors. So you need more coffee in order to accommodate the increased level of receptors, and you have to essentially drink more and more. And then, there's all sorts of effects after if you stop cold turkey or if you're kind of tried to waning down and so on. Next
one, sunscreen.
Sunscreen.
Tell us the biochemistry of sunscreen, because I know there's the natural sunscreen, which is basically just 100% zinc you can put on your skin.
Like the really like white stuff.
Yes.
It's like really
the classic, sunscreen on the nose at the beach. It's all zinc.
I mean, like you said, there are two kinds there, and they mainly, from what I understand, block UVB rays. So there's different types of UV rays. For example, UVA rays are more, I think they're a longer wavelength, and they penetrate the skin deeply. And normally they're more related to aging, like so the kind of the sun spots on your skin, the wrinkles, and so on. UVB rays are like the shorter wave ones that are more damaging. They cause the different types of skin cancer.
Yeah.
But when it comes to sunscreens, I believe most of them generally tend to protect against the UVB rays, if I'm not mistaken. And some will have a formulation that absorb the UVB rays. And you have like the ones that reflect it. So I believe like the zinc oxide and titanium oxide ones, those tend to kind of like reflect the UV rays. But I don't know the exact kind of like time frame, but usually you should, if you're spending your entire day at the beach, for example, you need to apply it multiple times a day.
Yeah. Right, let's go back to just the science communication now that we've gone through those two little fun segments. How important do you think is it for communicating science to just the community and the public?
I mean, I think it's very important. Like I was saying earlier, that this kind of idea of the podcast started just because I was interested in people's kind of careers and paths and so on. And then throughout the later stages of the podcast, while I was still interested in people's careers, but it developed more into, I think that the level of communication here that I am helping to kind of spread the science and the work that they're doing, I think I can improve on that because communication, essentially, we as scientists are trained in doing the scientific work, and I mean, we do train in communication where we go to conferences, but that's not the kind of level of communication that you would need to kind of make videos or kind of content to the public where you're explaining very complex science in a more accessible way. So I think to do that, it's important to communicate it because it allows the people that aren't maybe scientifically inclined or aren't very interested in it to understand it more and to learn more about it and maybe develop more of an interest to it, whether it's for their own benefit, for health effects, or for people around you. It's great because the most important thing that science communication can help with is avoiding misconceptions and miscommunication when it comes to science, like vaccines are bad, or things in science that we know work and are very important. Good science communication can help kind of ameliorate all the bad negative side effects from it.
So now, obviously, with your work in the podcast and obviously your work in the lab, there's a lot of time and effort that goes into both. Which one would you say is harder? Would you say running an experiment in the lab at 2 o'clock in the morning that you need to complete, or trying to wrap up, the fine tuning a podcast at 2 o'clock in the morning? Which one would you say is more mentally straining?
Definitely, I would say it has to be the experiment.
Okay.
I mean, most of the time when
So
I'm
you're
doing
saying podcasts are easy.
Oh, absolutely not. Absolutely not. But I mean, like when I'm editing, for example, an episode, and it has happened when I'm editing, like at 2 a.m.
Yeah.
I'm usually, either at home, usually almost always at home, but usually at my desk or like on the couch, just kind of laying down and just edit it.
Yeah.
It's a bit more relaxing. Whereas I'm in the lab at 2 a.m. It's, I've been walking around all day. It's been a long day. I'm tired. I'm exhausted. I'm just trying to focus on not making mistakes and trying to make sure that everything I'm doing is correct. So, I mean, that, the 2 a.m. in the lab that was back during my grad days no longer happens anymore, of course. It's more of a 9 to 5 or like 10 to 6 kind of schedule now. But yeah, those days where, we're crunching numbers, trying to get the next result, trying to kind of, fine tune the experiment. Those were, definitely more challenging, I would say.
What advice would you like to give students who are looking to combine science and storytelling?
The most important thing is that you're genuinely interested and care about the topic. I think an audience can usually tell when you're not passionate about something, and it kind of detracts from the message.
That's fair.
So if you are trying to communicate certain research or science, I'd say make sure you're interested in it, because it also extends to you doing better research, more in-depth kind of analysis of what you're trying to communicate. And people can pick up on that. Like they can tell that you really are interested in that thing, and that's contagious to a degree, because they'll be able to remain more interested in it and engage with it better.
So where do we see George in the next 10 years? Do you think you would be doing more on the communication side of science, or do you see yourself more immersed into the experimental research side?
Oh, that
Tough question.
No. That just hits on like the kind of sore spot that I have about this, because right now I'm mostly doing research, and I find it very difficult to prioritize some time on my off time in order to do more of the communication as I've loved to do. Because as you know, I kind of stopped doing the podcast, or paused temporarily, I'm going to say, towards the end of my master's, because it was just so busy at the time. And I wanted to graduate on time and kind of get engaged at the time, which I did. So a lot of things going on, and I decided to put it on pause. I would like to, in 10 years, have a steady level of science communication where I'm more in the science communication side than I am in the research side, because I think it's important. I love doing it. I truly do. And I'm going to make mistakes, and I'm going to, going to get some things wrong, but that's just part of the process. And I would love to get back into it properly.
Yeah. Awesome. Well, again, thank you very much, George, for coming and joining us for See You at Kanata Talks. Appreciate it. You really highlighted how you're working hard to bridge the divide between science and communication and make it more accessible. So we really appreciate the work that you're doing, and we're looking forward to kind of following your career and seeing if the podcast can pick up where it left off, and seeing where things go with your work with NuvoBio as well. So thank you, George, for coming. Appreciate it, and we'll keep in touch.
Thank you so much.
Thanks.
Pleasure again. Take care.
Thank you for joining us for our See You at Kanata Talks. We hope that you were able to gain a little bit of insight of some of the skills that our students are learning here in the classroom, and how they're applying those directly, whether it's in clubs, whether it's in industry, or just on their own little research projects as well. With that, we want to invite you to reach out to us at See You at Kanata. We're here to help direct you to students, create that talent pipeline, or even to partner with us on a research capacity as well. As we may have learned from some of these sessions, there is so much that the university can offer, and we're here in Kanata to help you access that as well. So thank you for joining See You at Kanata Talks. I'm Travis Wheeler, and we hope to see you at Kanata.